So good morning, everybody. Hope you can hear me. My name is Anthony Kong. I'm a reader at King's College London. Has a special interest in the combination of radiotherapy with immunotherapy. So we have a speaker this morning, Dr. Fulvia Vascotto. She's the head of Vaccine and Cellular Immunotherapy unit at TRON and Mainz in Germany. I'm just going to talk a few things first before I introduce Fulvia further. So RadNet, for people who might be outside RadNet City of London, is obviously formed within City of London of the four institutes: University College London, King's College London, CRIC, and also Bart's Cancer Institute. So our RadNet series, seminar series, is a monthly series. We intend to use this monthly series to initiate connectivity between each other and to give insight to new radiation research, could be both clinical and non-clinical, and also to give the opportunity to build expertise and also collaboration with each other. So just a few things about this meeting, just a housekeeping rule. When the meeting starts, when Fulvia starts presenting, I'll ask everybody to mute their microphone and turn the camera off. And if you've got any questions, please write it down and then you can raise your hand during the chat. Fulvia said that if there's any burning questions, she's happy to interrupt her talk and answer your questions if you think that is necessary for her to carry on. So I think she's happy to do that. So I'm just going to talk more about Fulvia. So Fulvia worked at TRON, as I mentioned. So TRON actually was founded by Ugur Sahin, who also founded BioNTech. I think people are more familiar with BioNTech because of the recent RNA, the COVID-19 vaccine. But actually, TRON worked very, very closely with BioNTech and has been pioneering novel platforms for individualized therapy and biomarker, translating basic science into drug application and for clinical use. And for that purpose, they work very, very closely with BioNTech. And many of the previous RNA vaccines were discovered and found at TRON and then subsequently collaborated with BioNTech for clinical translation. So with that context, actually, Fulvia actually went to TRON and has done amazing work there. So before she went to TRON, she was a postdoctoral fellow at INSERN Institute of Curie and then Harvard Medical School. At TRON, Fulvia actually pursued a project on characterization of tumor microenvironment, adaptive T cells, and innate immune response, and also looking at the mode of action of several combination cancer therapy in murine tumor model. And I had the privilege of working with Fulvia at TRON, where we bring the animal irradiator to TRON, and it was really stimulating discussing with Fulvia, really, because she's always so energetic, full of enthusiasm about radiotherapy and how radiotherapy could prime the immune system. And it's through those discussions I had with Fulvia that also shaped my own research direction in both clinical and preclinical area. So we're really excited to hear Fulvia talk today, and she will talk about the good, the elder, and the ugly RNA vaccine and the radiotherapy to fight cancer. So really look forward to her talk. So Fulvia, I'll hand over the time to you, okay? Just remind everybody to mute your microphone and also switch off the camera when she's talking.
Okay, good morning to everybody. Yeah, thanks again. Just a moment, sorry. Anthony, to the kind introduction. And indeed, is really my pleasure to have spent the time with you in TRON, where it was an exciting always discussions among your expertise as a clinician. And I define myself a strong immunologist background. So I was really happy to, I'm sharing the video, right? Everybody, can you see Anthony?
Yes, we can see, yeah.
Yes.
Can see.
I'm doing the presentation mode shortly. Yeah, here we are. Just to make a bit of fun of or remind the good aficionado or interested people in the cinema, but of course, like I am, and Italian old film, and so it reminded me definitely that the approach that we have having in TRON was the good one that now we have learned from BioNTech translation how important was. And the elder, of course, was the radiotherapy. That is, you are the expert, and so I'm really happy that I could have your feedback on the approach that we selectively use in our preclinical study. And of course, we want to fight the ugly, that the cancer was the major aim of RNA platform development in TRON and then in BioNTech. So, right, I will present in 3 slots today some data. First of the platform, briefly of the RNA-LPX as a genetic vaccination platform that we developed combination with the checkpoint inhibitors, and then at the end the core part where we synergistically find a mode of action of local radiotherapy with the LIPO-MERIT. So I would present you shortly an historical slide that allowed you to understand how far ago, let's say, all this RNA and mRNA individualized immunotherapy started, thanks to Professor Hugo Schahin, of course, that he was the founder of TRON in 2010, and then he was already professor in Mainz, and so these reflect how long we try, and I will describe you lately all the process from the preclinical, but of course also major interest from patient to bring to a product and to cure and to translate in clinical applications. I enter in the detail a little bit because the IVT-RNA design was something that was very useful to as a molecular biological approach that is very easy to produce and purify, very easy to modify, and no integration into the genome because it relies completely on the cytoplasmic localization of the RNA, and has an independent HLA restriction because the patient or the treated patient exactly produce the protein and present the epitopes on the HLA endogenously that is carry on. And then, of course, the aim is to from the induction of the on the T cell immunoresponse. So what I have, I think it's here, yeah. I don't enter now so much into the detail because, of course, all the work that was published and was really enhancing and improving the stability of the RNA, and then for the different component depicted here. The most important overview that I wanted to give you and the concept is that we had developed 2 types of mRNA, or not only BioNTech but also other company who works in mRNA in the world. Are mainly based on the mRNA that is called immunogenic, and this was the one that I will talk about mainly today with you. And an immunogenic relies because it's an adjuvant effect, so it's heavily recognized as a mRNA by the cell and immunocell especially. And then we have the non-immunogenic mRNA that we don't want to talk about today, but we know the benefit since it's implemented in the COVID-19 vaccine, where it's less immunogenic, but they're definitely able to translate the product that you are interested. And of course, the application of the non-immunogenic RNA is not only for protein for vaccine as in infectious disease, but also has been used for protein replacement. So again, another little overview for the benefit that then mRNA vaccine have shown during this year. First was the intranodal, so the local targeting of dendritic cells, but intranodal injection. And this was an old approach that we also perform, TRON perform in collaboration, of course, with BioNTech in a patient. But then thanks to the development of a very important formulation, the delivery of mRNA, because need to be protected, of course, in the bloodstream if injected intravenously or in another route, encapsulated in some specific particles, allowed to be to target specifically the dendritic cells. And I will give you a little bit an overview of that. And a last concept that allowed you to follow the presentation today is more the approach that was designed for the neoantigen discovery that we call individualized vaccine for individual therapy for cancer patients. And here is nicely described that, of course, we take the sample from the patient. We could do the discovery of mutations by next generation sequencing mainly, and also RNA sequencing. And then, thanks to this in silico, then approach with the neo-epitope prioritization, that is still a very important task for bioinformaticians that invest a lot of time to improve that. And then the selection of the target. And of course, then we have the template that we need to produce then based on the neoepitope, the neoantigen that we design and we select, and then pass for the GMP production and of course the application to the patient. So this is indeed the platform that it took, I would say, 6 to 8 years to develop and to validate and to come to complete formation, complete validation of the platform from this lab to the patients. And it was an amazing achievement that mainly BioNTech was able to do as for the clinical translation of concept, that TRON was involved more on the research discoveries. Here, since I'm an immunologist and involved in cancer in the last 10 years, this is something that is really a milestone picture that everybody, I believe, has seen, and allowed you to understand how immunocells, the immunocell cycle, is involved in an immunoresponse. And this is something that, of course, you could see that is a lot of steps where peripherally, for the first time, the tumor grow and then could release antigen. This could be in different forms. And then the dendritic cells are the cells that are on the periphery, are the one able to migrate to the lymph node. This is known by ages by the immunoresponse and vaccinology also, because those are the migratory cells that are able to bring antigen from the periphery into the lymph node, where the immunocells, the immunoresponse start and generate. Here the dendritic cells encounter the T cells for the priming, and then here an activation to allow them to proliferate and then to circulate, to able them to reach then via bloodstream the tumor. So this is a cycle that is very important recently for the cancer, because a lot of steps were in I can put a pointer, sorry, I didn't do before. Sorry, a minute. Here, yeah. Sorry for the delay. So this is the components that are involved in the checkpoint inhibitors mainly, but also chemotherapy and radiotherapy to induce the tumor cell death, and so on in all the steps as depicted here. The most important that I was eager to be part in TRON during the development of cancer immunotherapies and combination is when combining how you organize the combination therapy, dose, timing, route, and of course taking in consideration the side effects. And this was a very nice collaboration with BioNTech, of course. Here I wanted to depict differently what then we aim with our RNA vaccine, that I will show you some results, but just a picture that allowed you to understand that the vaccine reached the spleen of the in the preclinical study in mice, but also in human. We will see a bit of the data. And these data reported here was able to demonstrate that the RNA vaccine can induce priming of T cells at both subsets CD4 and CD8 to allow to mount an important and complete immunoresponse. I will talk today definitely with the two parts, also activation of CD4 and activation of CD8, or in the mixture setting. And we will drive you step by step. But just to keep in mind the setting that the RNA vaccine is able to selectively, specifically induce one of the other or both. So the work was generated for a very talented PhD student at that time, now scientist and in BioNTech, Lena Kranz, with the director Mustafa Dicken. And that is the first one of the first paper where we described the delivery of the RNA vaccine by the formulation. Since it's published, I don't enter into the detail, but this is just the most important slide that was able to understand how formulation, that is from the BioNTech side, helped enormously to find the best charge ratio of the lipoplex, so RNA complex with a lipid. So this RNA lipoplex was charged in a different way that from the positive to the negative could be delivered specifically to different organs. And as you can see here, heavily positive was on the lung of the mice. This is a BLI readout, of course. And then in the more negative charge, the particles start to exclusively target the spleen, as you can see here. And this is the particles that we selectively and constantly use for cancer immunotherapy in preclinical studies and in humans. Additionally, also lymphoid organs was also addressed, like bone marrow and lymph node. And this is the example in human, where indeed the lipoplex for metabolic measurement was able to evidence metabolic activity in the spleen exclusively. So I enter now more in the detail of the immunoresponse that we were able to generate, and this is an example. We will go more into detail lately for the tumor model, but this is a E7, so the protein encoded by HPV16, that then was one of the tumor model antigen that we used to induce immunoresponse, and of course is a very immunogenic protein because it comes from a virus. And then we have tools to follow the T cells that are exclusively specific and able to recognize a peptide for E7. And we can follow in time how the T cells increase in time after two vaccinations. And it's a very important aspect of immunotherapy to induce CD8 T cells are able to kill your target. And we will see that. So this is the first example that was published in the Nature paper that we addressed the TC1 tumor model, that is a mouse model of head neck cancer expressing E6E7. And then here you can see that if you start at day 7 the therapy or at day 10 a bit lately, notice the scale, you have a very important tumor rejection on the mice. And this is done with the triple RNA vaccination treatment. And was really important and obviously very encouraging, but this was a very easy tumor cell type to treat with RNA. With another more challenges was CD26 that carry also another antigens, but here we use a CD4 T cell model epitope, and this was definitely also the case. And then this is another interesting approach, but because again, resuming the strategy of viral antigen, this is a GP70 peptide that comes from viral strain integrated in the genome of mice, and this is a very useful model and tool to follow CD8 T cell response. And again, as you can see here, the mice that are treated with the GP70 specific RNA tumor model are able to completely reject the tumor, while the control in the tumor grow and they die for that. So the last example was also in melanoma, that for another CD8 model antigen, we were able to cure indeed the tumor-bearing mice. So all this was encouraged in clinical study, and then in that work, we also share data that for the clinical study that was running at that time, and this is an ELISpot where we can follow the T cell reactivity, mainly for the antigen reported here, of course, neurocasone and mix of peptide control. What is our control settings? And then you can see that in time, based on the several vaccinations that the patient was having, they mount a potent T cell immunoresponse. And was very encouraging to see, indeed, of course, circulating in the blood of the patient these specific T cells after the vaccination. Based on this data generated a while ago, of course, two PhD students join sequentially the project. I will show you the work first that Christian developed as an immunotherapy, of course, and then lately Nadia developed for in combination with the radiotherapy. And again, this is the scheme that I showed you before, just to remind that then we focus specifically only to the CD8 RNA vaccination, so epitopes that are specific only for CD8 T cells. And here is the sample that we showed previously, and this is the immunoresponse that we were able to follow. And nicely, as we can see previously, but also for a very late therapy where tumor were very well established. So 2 weeks almost after tumor implantation, we start the tumor. We have a great immunorejection, but unfortunately, if we stop the RNA vaccination, the tumor relapsed. But was really promising in times and as depicted by the survival, that indeed you could have a survival benefit linked to the early treatment, of course, of RNA therapy. And we were more interested to stick on this scenario, of course, because as you know, more than me as a clinicians, a patient coming to the clinics already with unfortunately very established tumor. So it was a challenge not to work on this setting that was really easy, but then stretch the condition and challenge the system for the HPV tumor model, but the tumor was very big. And that's why for the next slides I will show you, the setting is always based on the late vaccination. This characterization of the tumor that we could see not only in DC1 that express only 67, but also on the C3 tumor model that express more viral antigens. We were able to induce T cells and then strongly induce T cell recruitment intratumorally in both tumor models. This is then, in addition, a very important slide that to show immunologically that the tumor the vaccination, can induce a memory response. This was an example where we took the mice that were surviving after the previous vaccination when we start at day 4, and then we were survived, all of them. And then when we took those mice and then we re-challenged them with a new tumor injection, all of them survived, indicating a very potent anti-tumoral T cell response mainly. And in the paper, we showed that it's CD8 T mediated. Another scenario that we were interested, of course, as I told you, the challenging situation is that we were perform a very late RNA vaccination, but unfortunately, the tumor relapsed. And when we try to understand what was going on those tumors, on the by RNA sequencing of the primary tumor or by histology, we were able to show that indeed a lot of checkpoint and the inhibitory molecule was involved. And unfortunately, then the tumor, like for example PD-L1, was so heavily overexpressed that then the T cells still present in the periphery and to the tumor were not able anymore to fight and react to reject the tumor. And as you can see, the tumor relapsed. So we were, of course, interested to translate then in how we could treat those animals. And then, of course, since the readout and the ongoing known powerful tools of anti-PD-L1 antibody, we combine this antibody on the RNA vaccine. As you can see here, if the E7 RNA, we perform also in addition only one vaccination and not 3 as previously to make room, of course, of window of time in the preclinical studies for the combination therapy. And that's why we were running on a suboptimal setting the RNA vaccination, so bringing only one vaccine at the injection at day 16, but then follow up by the treatment of anti-PD-L1. And as you can see here, was a strong benefit of the combination therapy. So again, as I said, that all these preliminary data allow them to generate the new PhD program of Nadia. That was a very talented student that completed the program last year in August, and she combined the radiotherapy to this setting. I don't need to recall to you that radiotherapy recently and are involved strongly in the immune response. And again, I want to highlight that, of course, it induces also an immunocell cycle. And this is why immunologists were very interested to show and to understand mainly how this induction of immunogenicity can be generated in different tumor models. As I said, we combine in them with the RNA, but the study that Nadia focused mainly and was already published was in a combination with RNA, not only for not for the CD8 epitopes as described before, but in a more challenging setting like the CD4. And to depict a bit more and to understand why the role of CD4 in tumor, I go back a little bit on the previous paper that we published in 2015, where the role of CD4 in the anti-tumoral immune response is really dominant and very important. So I described to you that at that time, as in human now for clinical, we took the sample from the tumor of the mice, the tumor cells mainly, and then healthy tissue. We compare the genome to identify in the tumor cell lines mutations that are carried on those preclinical cancer models. We may focus on CD26, B16, and several tumor models were sequenced, and then by the mutation prioritization, we can generate a priority list of neoantigens to induce immunogenicity tests that we evaluate in vivo in mice, which are among the mutations that has been found in silico, which are the one able to induce an immune response. And this is a test that, of course, was, as I said, published, but was allowed and for the work that Nadia focused in CD26 tumor model to concentrate in one mutation, as depicted here, that we call mutation 90, that is inducing only CD4 T cells, as depicted here. So this is just an example, of course, in a very early treatment with the several RNA vaccination that if we inject CD26 tumor cell lines that express luciferin as a reporter gene for the bioluminescent readout, you can see that then control mice have a heavy number, a very high number of tumor nodules in the lungs. This is a metastasis model that we call. While the mice treated with the RNA, and this time is called pentatope 1 and 2, that include several epitopes at that time, but include only also CD4, they were able to completely protect the mice for tumor burden. And this is why Nadia took part of this neoantigen, and mainly that we call PME, that involve almost exclusively this mutation 90 that trigger only CD4 T cells. And she was interested to see then in combination with the radiotherapy how CD4 T cells triggered by the RNA vaccine can protect the mice from the tumor growth. And indeed, this is the case. This is the legend, just a color code. You could see and focus on the control RNA, that is an irrelevant RNA with the radio only, and the radiotherapy has a very tiny beneficial effect on CD26 tumor model. And then, of course, then in the combination where the RNA is depicted here, this is acute model. The RNA show a very almost no effect, as you can see in survival, because in the intratumoral, in the subacute model, it's very difficult to treat these mice. And as I as you can see also, the treatment of the RNA was not started so early as before intentionally, but just at day 10 with a repetitive weekly vaccination. And this is then the synergism that you can appreciate when we combine RNA-CD4 RNA vaccine with a radiotherapy, with a very nice benefit in survival and rejection of the tumor. What I wanted to point out also is the beneficial protocol that we select. Of course, for us was very important first to generate the CD4 T cells to be able to circulate into the mouse, because of course the induction of the vaccination RNA needs 7 days, of course, to induce an important and detectable T cell response in the blood. And so our aim it was first to generate the CD4 T cells in the window of time that then the tumor are not too big and too difficult to treat. Then we implement the radiotherapy, and then we follow to another RNA vaccine to boost. The interesting part is to focus on the RNA right now, because of course of the radiotherapy right now, because in this tumor model we were very interested to understand how this synergism occur. And this is one insight first. So these are the blood in the circulation of in the blood of the mice, and we can find the circulating T cells that are specific for this tumor model, as described to you before. And then, as you can see, radiotherapy itself can generate in a not very early, but 10 days after radiotherapy, we could detect an increase of the T cells induced by the only radiotherapy. And then this frequency and the circulation of the T cells were enhanced by the RNA vaccination. But the RNA vaccination is not with epitopes of CD8, okay? It's only epitopes encoding epitope for CD4 T cells. And this is the nice effect to show that the synergies occur. So the radiotherapy would generate in this tumor model a little pool of CD8 T cells, antigen specific for the tumor, and then the T cells would enhance and help them. And this is the concept of licensing and help of the CD4 in the immunotherapies. We dissect more and understand more the tumor environment. So we repeat experiment and we perform readout at day 29, so at around 7-8 days after the radiation, and we with the fax analysis that are really powerful to detect many cell types, we could see reduction of the tumor cells in the radio and in the combo, and then as well again the pool of CD8 total, as well as in the tumor of the GP70 frequency of the GP70 specific T cells as depicted here. So this was very promising, and then we wanted to understand a bit more the effector function of the T cells, because at the moment we noticed frequency that was much higher in the tumor environment. So we took the mice, we repeat the experiment, we took the mice and we phenotype more the T cells that are intratumoral. So all this data comes from analysis of T cells that reside at that time in the tumor of the mice. And we can find that, of course, in the chemotherapy, but the most known PD-1 inhibitory molecule was really down-modulated on the T cells, as well as LAC3. It's also known to be an inhibitory molecule involved in the T cell response. And interestingly, with the intracellular staining and in vitro restimulation of the T cells with a specific peptide, we could follow the reactivity of the T cells. So with a specific peptide, as I told you, this is a sort of tumor-associated antigen. We use this as a model. We can load the target cells and then check how much T cells, the reactivity in vitro of the T cells specific for the peptide. And as you can see, radiotherapy was not able to do so much at this time point in terms of reactivity. However, in the combo therapy was really, really strong. Good. This was the consideration. What also we were interested based also on the always clinical translational aspect of local radiotherapy was to understand if the single dose versus the fractionated dose showed differences in this tumor model. And we decided to take biological effective dose equivalent to the 12 gray, so we decided to give the 3 times 6 gray. Of course, in the mice, the patient schedule is very difficult to follow because the tumor um, the tumor growth is very quick compared to the patients, and that's why the completely clinical protocol is almost impossible to dissect and to treat in mice. However, we wanted at least to try what was known in literature that around 6 or 8 gray was shown to be really important and better of the higher dose as a 12 gray. And in this tumor model, actually, what we noticed that the two treatment was not differing so much compared to um, the other studies in other tumor models. And so in this way, the RNA vaccine was conducted in 3 times as previously, and again the irradiation was done in a single day here for the 12 gray, and then we didn't notice any differences between the response of these two treatments in mice. And then we could follow also that both regimens allowed a very nice induction of CD8 T cells specific in circulation into the blood, justifying the biological effect and the biological anti-tumoral effect that we saw in the mice. So what we came also in another, of course, as immunologists, we always were interested to see, okay, in the previous experiment, as I showed you before, some mice were still alive, so tumor free, and we wanted to test them if they were able to really be protected to a second tumor challenge. So if we were able to have memory for this tumor model. And what we make usage at that time was also to a cell line that was generated to express to not express the immunodominant tumor-associated antigens that we were describing so far. So the wild-type cell line was injected before to generate this survival curve, but then we re-challenged with a tumor cell line that was lacking this antigen, understanding if other T cell epitope was also developed. And this is the case indeed, that the mice were all of them able to protect. Both are reported on the top here, and for the pool of CD4 of the single CD4 RNA epitopes, and both of them are able to, so means that the CD4 T cells that we induce with the RNA vaccine are able to help the CD8 antigen-specific tumor response that is induced by the radiotherapy in a immunodominant independent manner. So this GP70 antigen called as immunodominant was not the only one able to drive the antitumoral response. And then this make the interest to go on the synergy on another tumor in the same tumor model from the beginning, where we don't have from the beginning these immunodominant epitopes, this immunodominant antigen, and we perform the treatment as described before. And as you can see again, if the RNA and if the radio induce a slightly benefit of survival on the tumors. The interesting part was to have the combination that allowed a very good survival of the benefit in the therapy. Here, we were not able to have, unfortunately, any CD8 epitopes, but we follow the rejection in vitro of the same tumor cell lines. And as you can see, radiotherapy induced mainly reactivity of these cells. So we follow the interferon gamma secretion on these tumor cells in the radiotherapy only, as well in the combination, as well as if we irradiated the tumor cells in vitro previously. Okay, so then I think this is the last part. The interesting approach, of course, for additional translation interest on these therapies was to combine with the checkpoint inhibitors. And what we decided for this tumor model was to combine anti-PD-1 and anti-CTLA-4. So the protocol, the scheme, the experimental setting is defined here, very similar to the previous one. So always RNA first for CD4 to mount and then generate a CD4 T cell response specific for the tumor, and then in combination with the radio. So here are all radiotherapy treated mice because the experiment otherwise would be super big. So we decided to do the triple treatment versus the double treatment the analysis. And so in this way, all the mice here are treated with radiotherapy, but then of course, or treated with anti-PD-L1 with a control RNA or with the tumor-specific RNA. And here is nice to define that the benefit of the triple combination therapy versus the dual combination therapy is very, very important and very promising for the translation in clinical. And again, able as immunologists to follow the T cell response specific for RNA and the tumor model, we make usage of the interferon gamma ELISpot, again, using as a target the tumor cell lines. And we were able, again, non-irradiated or irradiated. With irradiation, we mount this immunoresponse in terms of intensity, but was really very important to see that the radiotherapy in this tumor model generated in combination with CTLA-4 has known a very important immunoreactivity as well as in the triple combination. Okay, good. I think I reached the end of the talk. I'm happy to tell you and to have shared with you data to show how the RNA lipoplex vaccination expand strongly antigen-specific T cell in preclinical models and in human. The combination with checkpoint inhibitors, as example anti-PD-L1, but also anti-PD-1 and CTLA-4, always achieve superior anti-tumor efficacy in the tumor models. And then the RNA lipoplex vaccine encoding CD4 and CD8 epitope. For the CD8 epitope, I didn't show you yet, but in combination with the local radiotherapy, synergize in a very promising manner. And this is, of course, the most important aspect for translation in this setting was also to find a very successful setting in preclinical to find the synergies in a triple combination therapy. Okay, with this, I want to thank all the people involved in this work. Of course, mainly Ugo Shahen and Özlem Tureci as a founder of this exciting group of works that expand from TRON to BioNTech, and then people actively discussing the data, and as Sebastian and Mustafa, and of course the executor like Nadine and Christian Gurwitz and Lena Kantz for the data that we generate. And thank you for your attention, and I'm happy to have questions now.
So thank you, Fulvia, for the excellent talk and very comprehensive coverage of your work. So we will open to questions. So if people are shy about asking questions, you can also type the chat, and then I can relay the questions to Fulvia for you. I will start by asking a few questions first. So Fulvia, I just wanted to, first question is more for clarification. Initially, when you showed us the HPV vaccine, it doesn't seem to induce sustained response on its own, because in the late vaccination, the tumor started to grow. But you then showed that it can still have a memory T cells because it rejects the tumor. Just for clarification, I might have missed something. So how does that work if it doesn't have the sustained response, but it can still have the memory T cells to reject the tumor when you re-inject the tumor into the mice?
Okay, so for the re-challenging model, I mean, this was done. So this was the setting. The latest you vaccinate, the lower protection you have, as the depicted here. So for the rechallenge tumor model, we took a setting where we start pretty early. So most of the mice were tumor free, or were able to reject the tumor. So they see the tumor, they induce the immune response, of course, also by the RNA vaccine, and then they were able to to reject the tumor. So they were completely on the setting to be tumor free. Then come the rechallenge, and that's why since they are tumor free, we re-inoculate the tumor cells after a while, and then I can go back to that slide perhaps here, and this is the case where all of them after 100 days that were tumor free, they were re-challenged with a new tumor, and then this is an evidence that then they mount an immune response. So in this setting, since we vaccinate is, let's say, obvious, but of course was a very important milestone experiment to show that then the vaccination induced a memory T cell pool. But in addition, in the combination therapy that you perhaps also refer, because we have the rechallenge also there, here were the situation that indeed here we don't do we did vaccination for CD4, but equivalently well they are able to mount a memory response in the same way.
Okay, so on that same note, there is a question from Jamie Dean. He said, thanks for a very interesting talk. In many of your experiments, a subset of, but not all mice had complete responses to therapy. Have you done any work to try to understand why some of the mice has complete responses, but others don't?
Right. Let's say it could be that we don't know. We never did any individual analysis of mice that did not respond, like on the treatment. However, it's an interesting approach that obviously we could consider. We consider that overall the survival data that we show was really significant and very helpful. But it's a case then the model also we also need to. I need to highlight that all the therapy are done in a way that we synchronize. Let's say we randomize the group. So in terms that at the day of the therapy, we need to take mice in the game that have a certain tumor mass, and all of them need to be in a definitive range. When we do that, we exclude mice that has not been engrafted or grow too quickly. So after this preliminary study, that's why the number of mice implemented in this experiment are really big. Then we are sure that most of them are in the same time characteristic of tumor growth. But nevertheless, indeed, some of them did not respond completely. We are showing the data of the average, of course.
So Fulvia, I'm interested in the way you schedule your vaccination and anti-PD-1, anti-CTLA-4. So you sort of do the radiotherapy in the middle of vaccination after 2 or 3 vaccination, and then your anti-PD-1 and anti-CTLA-4 sort of in your subsequent experiment sort of introduce sort of afterwards as well. So my question to you, I think many people in the audience probably would be interested in your rationale for scheduling.
Yeah.
And whether if you schedule differently would have make a different result. For example, if you have treated anti-PD-1 or anti-CTLA-4 early on rather than later. And also, obviously, triple combination is more complicated. Scheduling with the vaccination or with the radiotherapy, how you decide the scheduling will be very interesting for us.
So I have to say that the first rationale for us was to have, so we know that the RNA vaccine need approximately 7 days, as I said, to mount a good number of T cells able to circulate and then to reach the tumor. So this is the first things. And most of the checkpoint, in this case, PD-1 and CTLA-4, are targeting T cells. So the rationale was, in this case, to have the circulating T cells first. That's why we initiate the RNA vaccine. But then the radiotherapy, as we described, is able to mount to induce tumor cell death. We didn't characterize. Is other study in literature showing much better, at least for chemotherapy treatment, how this tumor is very sensitive. So we consider the radiotherapy here is an in situ immunization. So indeed induce cell death that then the cells can be uptaken and then start the immunocycle. So that's why we saw the CD8 T cells in circulation after radiotherapy. And so this is again something that this loop of induction of T cells need also a certain time, is not immediately, while the antibody is very fast. So you inject and they have the effector function immediately after injection. So for this rationale and kinetics and dynamic, we decided to go first on this setting. Also because, as I said, in this case, the anti-PD-1 and anti-CTLA-4 need to see the T cells. So first the Tregs, of course, in the case of CTLA-4, but mainly the anti-PD-1 to support the T cells that are induced by the radiotherapy. That's why the rationale to go in this setting. For the combination without radiotherapy for the PD-L1 that I showed you also in the previous study, but again, no radiotherapy, there was anti-PD-L1. The rationale was a little bit different because, of course, here, as I showed you in the immunofluorescence, the RNA and the gamma produced by the T cells that try to kill so much the tumor induce gamma on the tumor cell, induce PD-L1 on the tumor cells. That's why the rationale here was also to go after RNA with the anti-PD-L1 antibody, because first to induce the T cells, they act, and anti-PD-L1 could have then a good substrate on the tumor to dump the inhibition. However, I can tell you that we didn't test so many alternative protocols, okay, to prove that this is the most successful. We came on this rationale, we use these, and sometimes it's difficult to convince authorities to make an experiment in mice just to prove something for curiosity, of course. But would be important, of course, to dissect more protocols, but we didn't do it yet.
So do you have any questions for anyone else? You can raise your hands. I've got another question is, when you did the triple combination, you obviously use anti-PD-1 and also anti-CTLA-4. Have you noticed the difference that the graph seems to suggest anti-CTLA-4 seems to be better in the triple combination, or have I got it wrong?
No, it's true. And this now comes with a specific tumor model that we have here. So anti-CTLA-4 is, this molecule is mainly expressed on the Treg, and this tumor model that is CT26 is heavily enriched in Treg tumor infiltration. So that's why this tumor model have a tremendous benefit already without specific RNA in the rejection of the tumor. Yeah, but
Okay.
this is specific of this tumor model. Not other tumor models are so sensitive of anti-CTLA-4 like this one.
And Carol.
Hi, Anthony. Can I ask a quick question? Thanks so much for a great talk. Crispin Harley here. Just a quick question. I'm sorry if I missed it. Obviously you focused on CD8, CD4 infiltration, but did you have a look at what effect this combination has on other components of the TME, such as sort of myeloid cells at all?
Yes, in the paper we have done an extensive. Unfortunately, I didn't bring that part of the. In this study, in the monotherapy, we have an extensive tumor environment characterization as well, and in the CD4 on the macrophages mainly. On the myeloid suppressor cells, not so much in terms of the different subsets, but on the tumor, we show that here we promote apolarization to M1 on the tumor-associated macrophages.
Okay,
did I answer your question?
Yeah, exactly. So you see more infiltration from macrophages or less?
In terms of frequency, I think we didn't notice so much different in frequency, despite we always want to also validate that. We showed more that we have more M1, so INOS high and CD206 low macrophages in the combi therapy.
Okay, great. No, thank you very much.
You're welcome.
We're coming to the end. I've got one more question, actually, very quickly, Fulvia. You obviously chose 12 gray, which is very interesting to me because we don't use that dose in the clinics. And then obviously you show that the fractionation 6 gray times 3 with the equivalent dose is the same. But I just wonder why you chose 12 gray? You have used other dose before, and what is the rationale? Okay, just because for us who started experiment now, we just really sometimes dose is a factor for us to consider, and it's intriguing to see how you decide that.
So as I said, we were very naive at the beginning. You were inspiring us more on the different clinical protocols. I have to say that screening literature, we found that in this local radiotherapy protocol, people were using 20 gray in mice. And when we started as a preliminary radiologist, radioimmunologist, we wanted to see the effect on the tissue. And then we noticed for the H2X marker and all the effect that 12 gray was already very high. And then was always in other literature find that the 8 gray was the second option of single local radiotherapy. So at the end, I have to say that we didn't want to burn the mice with the 20 gray, so that we found extremely high, and also feasibility because we are in a preclinical model, and this already took 20 minutes for us to irradiate these mice with 12 gray. So technically, we find a compromised situation. However, I'm really favoring that always as a immunologist and the translation in clinicals that let's bring a patient less in clinic to a treatment, right? He will be super happy, and that's why I favor to think that it's better to be hard and rush once. And then see the effect as an immunologist. Sometimes it's much better than to be persistent and induce chronic inflammation. So this is just speculation. I don't have data to show this gut feeling, let's say, on the different regimens that we have done now in combination, and that's why we select. But it will be definitely interesting to see if this rationale in the preclinical studies would be important then for clinical.
Thank you. That's really good. Do I have any question for anyone else? If not, we will conclude this meeting. And we really, really enjoy your talk, Fulvia. It was an excellent talk, and we really learned a lot. And thank you so much for coming and to give us a talk on your work. And we really, really value your opinion and advice to share with us your previous experience. So thank you so much for coming.
Thank you for you too, Anthony, for the invitation, and everybody for attending the meeting. Thanks a lot.
All right. Everybody has a nice day. Take care now.
Bye.
Have a nice day. Bye-bye.